The central dogma of molecular biology is the idea that we go from DNA to RNA to protein. And by central dogma, we mean that when you're considering molecular biology, the very center of all our understanding is this idea that we go from DNA to RNA to protein. It's almost unthinkable that this does not happen. Now, as it turns out, there are exceptions. Like any good rule, there are exceptions, and for instance, some reverse transcriptases take RNA and go back to DNA. And this idea that all information starts with DNA and moves on to and finalizes with a protein, this was a big question. It didn't just come easily as an understanding. It took a lot of experiments. There were some really, really important experiments that I've already photocopied and handed out to you previously. And I asked you to read it and then to put it aside because we would get to it in chapter 7. And here we are. And these experiments were incredibly important in figuring out that DNA is the source of the information and not protein. Because initially, scientists were able to find a group of molecules called DNA and another called RNA and another called protein. And we couldn't figure out at first which one held the information that Mendel had described as the inheritable bits, right? Remember, Mendel didn't know what a gene was. No one did. But we knew there was this information that could be sent and could be inherited from parent to child. And we learned about how these, via experiments like Mendel's, we learned how these things could be inherited, how they were passed down. We even knew what cells carried these. And so we knew that we had sperm and we had eggs, but we didn't understand what these molecules were. How was this? What held that information? What was the stuff that was sent from one cell to the next, or from one parent to the next child? What form did that molecule take? Was it a protein that did that, or was it RNA, or was it DNA? Today we know it's DNA that holds that basic information and then sends it down the pathways to make a protein. Let me take a minute here to explain these drawings, although I hope you understand that already most of what you see here in the picture, I would assume that you could deduce anything you didn't know. But here we have our DNA molecule with our nucleotide bases, A, T, G, C, etc. Each hydrogen bonding to the other. We've talked about that before. That's DNA. And here it is in letter form, right? C, C, T, G. That's one strand. Of course, it'd be a complementary strand next to it, doing its hydrogen bonding. And we know that this long strand of DNA is read in it quickly makes an RNA molecule, and the RNA molecule there are no T's, so anywhere it reads a T, it's going to put a U, and it is an enzyme, and we'll talk about that when we talk about transcription. Transcription is the process of going from DNA to RNA, and where you see a T, where in RNA it'll be a U. There are no T's in RNA; they're only U's. Then you have, of course, your A's will be your A's and your C's your C's. So you see, here's a strand of RNA. Here's a strand of DNA. Now, for RNA to go and become a peptide, or a peptide, remember, a polypeptide is known as a protein, right? For it to become this polypeptide, the protein that runs the cell, because proteins do things. They are enzymes, and they are structural, and they're the machinery that runs the cell. All the other molecules are fairly supportive. Of course, we have membrane, lipids that create membranes that are very functional as well. But the proteins in the membrane actually do the function, don't they? Those integral membrane proteins. So proteins are incredible machinery that runs the cell and does the function of the cell. And without proteins, you just wouldn't be able to do it. But to make a protein, you need an RNA molecule. To make this RNA molecule with the right information, you need DNA. We know that the reason that you see these three-letter color combinations is because we call these codons. And it turns out that these codons code for an amino acid, which is what peptides are made of. If you remember back in a few months ago, we talked about proteins being a polymer of amino acids, one amino acid after the next, being connected with covalent bonds called peptide bonds. And these bonds, as you make one amino acid next to the other, the sequence of which amino acid, which of the 20 possible amino acids you put next to the one that you have, and which one comes next and which ones come next, that sequence can be found to originate in DNA. You can find it here in RNA as well, of course, but it originates in DNA. This sequence of which amino acid comes after the first one and the next one and the next and the next determines the shape of the protein. That shape, as we have repeated over and over and over again, that shape determines the function of that protein. So one specific shape, and you get a channel protein that lets stuff in. Another shape, you get a protein that only lets it in when a current runs through the membrane, an electric current. Another shape, and it only opens up if something bonds to that particular branch that changes the shape to allow the channel, the protein to create a channel where things might pass through. So when we're looking at the shape of the protein, it determines how it functions and what it does. That shape was determined by which amino acid is in what the amino acid sequence is, and before that is determined by the RNA sequence, and the RNA sequence is determined by the DNA sequence. So DNA is the beginning of the message. That's why we call this the blueprint of the cell, because it has the message that finally makes the protein that does stuff. If you're going to make insulin, you need a protein that's going to be able to catalyze the making of insulin, etc. So that's a central dogma, and basically, again, it's DNA to RNA to protein, and we'll go through and explain each of these. We'll talk about DNA. First, we'll figure out how did they know it was DNA. Again, I gave you that paperwork already. You should be able to read it on your own, but we'll make sure we'll go through that paperwork ourselves as a class. We'll figure out what RNA is, what is DNA, and what is a protein. Then we'll go ahead and discuss how do you make DNA, how is DNA replicated. How do you take the DNA and make it into RNA, this thing called transcription. And then we'll talk about how RNA is translated into a protein. So those are the four basic things. One is replication, which you don't see here. Next is transcription. The last is translation. How do you go from DNA to a protein? What impacts that process, and what's important about it? So that's our objective in this chapter. Okay, so this is the document that I had photocopied. You should have in your notebook. It's the transforming principle, identifying the molecule of inheritance. The first section we see here is looking at molecules of inheritance, and it's asking, which of the three, DNA, RNA, or protein, is the inheritable material? It talks about the Morgan fruit fly experiment, which we haven't covered and we're probably not going to get to. But in Morgan's fruit fly experiment, he noticed that the color of the fly's eyes were associated to a chromosome. At that point, they knew what the chromosomes were, that there were chromosomes, and that there was some correlation between the chromosome and the outcome of a mating. But in the 30s and 40s, they still didn't understand which of the three could hold all the information that's necessary to make something as complex as a human being. All the different organs, the cells, the colors, the shapes, the functions, all the different diseases that when things go wrong. It seemed like an amazing amount of information to be able to store in a cell. And so they figured, if you read here in this sentence, that most of the scientists thought it would definitely be proteins because proteins were the most diverse group of molecules out there. And so, therefore, if they're diverse, you would think, well, those could be the pages of the book. And the book has to be really big. So then, therefore, protein must be the molecule that was inherited, that held information. And they knew they had proteins in chromosomes. Remember, you had histones. So you know there were proteins. The question was, where was the information? Was it being held in RNA, DNA, or proteins? So there were a bunch of experiments that helped define this central dogma. The first important experiment that we have to talk about is this Griffith experiment, 1920. Gosh, man, it'll be almost 100 years since Griffith had done his first experiments. And you have to wonder, the first question they ask you is, why was studying pneumonia so important in the 1920s? I'll tell you this much, and this is a clue, some information that would help you answer that. So pay attention. In the 1920s, they didn't have antibiotics. All right. And the number one killer of people was contagious diseases. Not heart attacks, not accidents, not even war, but contagious diseases. All right, moving on. So when these scientists looked underneath a microscope, they could see that some of the pneumonia, right? Some of the pneumonia had these shiny coats. And because they had these shiny capsules, the capsules are made of these things called polysaccharides, which are a bunch of chains of sugars that create this real shininess, if you could think of like melted sugar, right? So this chain of shininess actually hid the bacteria inside this capsule and made it much more pathogenic, much more disease-causing, because your body couldn't react to it because the polysaccharide coating, much like the mucus in your membrane, the polysaccharide coating in this capsule actually protected the pathogenic strain. It looked shiny and smooth and caused disease because it was hidden from your immune system. The non-disease causing bacteria, the bacteria that wouldn't cause a disease, was rough. And you can see it here. It looks a little rougher, not as shiny. It doesn't glow as much. And they're smaller. And this bacteria here, because it didn't have this shiny coat, couldn't hide and therefore didn't cause a disease. Our body could fight off any over-reproducing bacterial colony and keep it at bay. Where in this one, if your body was attacking the bacteria, they had these coats that hid it from your body, so your body never attacked it, so it killed you. So Griffith decided to use mice to experiment on. And he started injecting bacteria grown on petri dishes and either made the mice sick or killed them or produced no disease, right? So if it produced a disease but it didn't kill them or killed them outright or it didn't make them sick at all. So those were the three options. And you see that there were two possible bacteria. Now let's see what kind of experiments he designed. So my hope is that you're able to do this on your own, but here you'll see, fill out the table below and diagram, below the diagram, predicting whether what you think will happen as a result of injection. Either the mice are going to live or they're going to die. Well, in the rough strain, remember, I'm not going to tell you, go back if you didn't hear it, but the rough strain I'll just go ahead and tell you since you're just going to rewind, was the non-pathogenic. So it wasn't hidden, your body fought it off. Is the mouse going to die or live? The smooth strain was the opposite of the rough. Is it going to die or live? Heat killed smooth strain. So this smooth strain, but it was boiled or come close to boiling. So it killed all, if not most, of the bacteria. Did the mouse live or die? Rough strain and heat killed smooth. So you killed these guys and you throw in some rough strain. Did the mouse live or die? Make your prediction. Well, there were some interesting results, and the results were rough strain, the mouse lived. Smooth strain, the mouse died. Heat killed smooth strain, the mouse lived. But rough strain and heat killed smooth strain, the mouse died. How did that work? What was going on? What information did allowed this bacteria to kill this mouse, even though the heat killed strain was already the disease causing strain was dead? Then somehow the non-disease coded bacteria learned to do something that killed the mouse. Huh. Now here's some really good questions for you that really you should be able to answer. Of these 4 experiments, 1, 2, 3, and 4, right? Which one was your control group? Which groups of these 4 groups? Which are the 4 groups that said, "Huh? Well, what if? What am I going to compare my results to?" And the second question is, which of the results is unexpected? I think you could answer that. And what are the 2 hypotheses that might explain the unexpected results? In other words, what might be going on in the system? So, let's go back and let me make sure you understand what's going on. Here's these rough strains that don't cause disease theoretically, and you inject the mouse and the mouse lives. It doesn't cause disease. The mouse does not die. Well, that makes sense. I'll help you out and point out that this is probably a what? A control group, since you expected this one to live. Smooth strain, supposed to kill the mouse. Well, you injected it and it killed the mouse. Again, another control group. This is your negative control. Here's your positive control. Heat killed smooth strain. It didn't die. Another control group, another thing to compare it to. And finally, your experimental group. Rough strain and heat killed smooth strain killed the mouse. So I guess, what are you trying to control in these 3 control groups? What do these 3 things help you compare your experimental group to it that helps cancel out possibilities? Well, the rough strain by itself did not kill the mouse. So the fact that this experimental group had the rough strain should not have killed that mouse. The smooth strain should have killed that mouse. But if I heat treated it, if I heat killed the smooth strain, then the mouse should live, since here's another control group that shows that when I heat treat it, the mouse lives. So there's the only thing that kills the mouse is not some toxin that's in the bacteria. It has to be the bacteria itself. The live bacteria must be the killer. But here we put in the dead material, the dead heat-killed smooth strain, and the strain that doesn't cause disease at all, and somehow it kills the mouse. So this is your experimental group. This is your positive, this is your negative control, and a positive control. What are the results that are unexpected? I think you can figure that out. I think you can write it out for yourself. What are the 2 hypotheses then that could explain this? How do you explain it? Well, let's think. Let's think about some things that don't explain these results. We know there's nothing in the heat-killed bacteria after they've been killed that would kill a mouse. How do I know that? Well, I have my control here, my negative control that shows that if I heat treat the bacteria, it's not going to kill the mouse. I know the smooth strain does kill the mouse, and I know the rough strain doesn't. So what is unexpected here, and why? And last, what are the 2 hypotheses again is, I guess you could argue that maybe some of these bacteria lived and that they ended up killing the mouse in the end. Well, that would make sense, since Griffith, I'm sure, repeated the experiment many, many times and made sure that whatever he did here that allowed the mouse to live, he did here. Another thing you might you might say that somehow this living bacteria makes some compound that reacts with this stuff and kills the mouse. That's one thing. The other hypothesis is a little more interesting, and that's that somehow this non-disease causing bacteria learned from the dead bacteria, picked something up here that it learned how to do. Some information it got from the dead bacteria allowed it to turn into a deadly strain. That's really interesting. So in the next set of experiments, Oswald, Colin McLeod, and McLean McCarty all started thinking that that information from the dead bacteria that could cause disease, that information must have been transferred to the non-disease causing bacteria, and somehow that changed its phenotype. Something in its genotype was changed. Some information was transmitted to the genotype of the non-disease causing to make it a killer. What was that material? Well, these guys came up with, they had 3 tools in their toolboxes, right? They had proteases, and I could have you look it up, but I'm not going to. I'm going to go ahead and tell you, because that's one of the first questions. What do they do? What do you think they do? Well, first is as the statement here makes clear, is that ACE means it's an enzyme. Well, it turns out, and write it in the spaces provided, proteases eat or destroy proteins. DNases, therefore, do what? They destroy DNA. And RNases destroy, you guessed it, RNA. So each one of these destroys any protein, DNA, or RNA in the system that you put them into. So if you have a test tube full of bacteria or materials and you add proteases, all the proteins will be eaten away. There'll be no proteins left. Just a bunch of amino acids floating in the solution. If you add DNase, then you destroy all the DNA in the test tube. So that only leaves you RNA. So based on that, go ahead and try to answer number 2. It's a little longer, and try to figure out how could you treat the dead, smooth bacteria with these enzymes, determine whether DNA, RNA, or protein is a genetic material. In other words, design an experiment using these 3 components that allow you to answer this question, whether it's RNA, DNA, or protein that makes up the genetic material. Well, here you have these results, and they're really, really interesting. It's a really elegant experiment. And what you have is this heat-killed bacteria. Now, remember, in the original experiment, you had the heat-killed didn't kill anything. The live, smooth bacteria did kill. The rough bacteria did not kill. But when you added the heat-killed bacteria, something in the what was left over of the dead bacteria, some information went from them into the rough bacteria and gave them the information they needed to kill. So the question was, is it RNA, DNA, or is it protein? Well, you were asked to design an experiment, and this is the design that they used. Again, it's very elegant. So you take this stuff, you add RNase. I ask you this: what is being gotten rid of with RNA? So you get rid of the RNase. Excuse me, I already told you the answer. Sorry. So RNase gets rid of all the RNA, leaving what? DNA and protein. So this particular experiment here leaves you DNA and protein. So I guess the question I have for you then is, what is RNA a possible? Is RNA a possible the material that transmitted the information that allowed this bacteria to be the killer? And I hope that your answer to that is something fairly clear. Is that a yes or no? Was it RNA? Is RNA the information transmitter? Did RNA make this bacteria a killer? Yes or no? The next question, of course, you get rid of here. You'll see here that you got rid of the protein. So that left you with what? Well, that left you with 1, DNA. And 2, of course, it left you with RNA. So here you had DNA and RNA, and guess what? The mouse died. So here we got rid of RNA, so we know RNA is not, right? Here we know RNA is not the information transmitter. But you do have DNA. And you have DNA in both this experiment and here in the first experiment. So there's the one thing that's common between the two that killed the mouse. So of course you have to do the third experiment, which is let's get rid of the DNA. So here we get rid of the DNA. So the only thing you have is one protein and the other one RNA. So you don't have DNA. And guess what? The mouse lives. Yay! So the mouse lives, and because the mouse lived, and you don't have any, what's missing in that particular experiment? There is no DNA. DNA must be the material that's changing the genome, and therefore giving the information to the rough bacteria to become smooth bacteria to show that phenotype and kill the mouse. Because when we get rid of DNA, in this experiment here, the mouse lives. In the other two, where only DNA is the common denominator, you end up with dead mice. I think a really elegant experiment. In fact, it is a classic experiment of biology. So this again was done by a group of scientists after Griffith. Okay, so here it's your task is to write an essay, a paragraph that describes the experiments done by Griffith. And of course, Griffith, Avery, McCarty, and McLeod. Avery, McCarty, and McLeod did the second experiment. Griffith did the first. Write a paragraph describing what happened. We just went through it. You can draw a diagram or a picture, but write the paragraph anyways. And if you do draw a diagram, you better make sure you're labeling it correctly and completely. Here, this next set of classic experiments in molecular biology was done by Mielsen and Stahl. And Mielsen and Stahl looked at some really interesting points of DNA. This is after we figured out DNA was definitely the molecule. The next question was how did DNA actually make a new molecule of DNA? So you had this long chain of sequences, right? CTG, ACT, TG. And next to it was the opposite. So you had two strands. You had the first strand was the one that had the information, the gene, A, T, G, C. And it doesn't have just genes. We know that. I hope you know that by now. And it goes on, for millions of base pairs. And then it has an opposite, what we call a complementary strand. So wherever there's an A, there's a T. Wherever there's a T, there's an A. Wherever there's a G, there's a C. And wherever there's a C, there's a G. So you have this complementary strand. The question is, how do you take this one double-stranded DNA molecule and make copies? How do you go from this to a new version? How does that happen? There were three options available. Either, as far as they could figure out, there were three possibilities. And the first was, it was semi-conservative. right? So that means that this strand, as they separated, this strand made a copy. So it had one new strand and one old strand. So it had the T, the A, the C, and the G, right? And then, of course, the old strand, still being in red, was A, T, G, and C. So the two strands kind of stayed together. You had one new, one old, right? New and old. So we call that semi-conservative. And this strand over here did the same thing. So you ended up with a T. You start with T, A, C, G. That didn't change, but as it opened up, you made a new one, which turned out to be A, T, G, C. And so you see there's one original strand in each of the two new duplicated molecules. Remember this S phase, the synthesis phase, where you're making copies of the DNA. You could have one old and one new. We call that semi-conservative. The next possible way that they could figure would be a method of making a copy of DNA is that you would end up with two old strands being together. So your A, T, G, C stayed together, and it had its complementary strand. The two old ones stayed together, and then you made a new one. A, T, T. Maybe that's how it happens. Man, maybe that's how it goes. So you had this old one over here. So this is the old. And then you made two new strands, and the two new strands went together, and the old one went together, and so that was a possible way. And then there was a dispersive where just little pieces and parts just went all over. You had new and old mixed in together in every part of biology, and all in every part of the DNA. Okay, so Maelson and Stahl used these two different isotopes of nitrogen, one being nitrogen 14, the other one nitrogen 15. This nitrogen 15 has one extra neutron. It's still nitrogen, right, because it has 7 protons each, but nitrogen 15 has an extra neutron. So it's an isotope. Two different versions of the same atom. Much like two alleles are two versions of the same gene. So here we're looking at chromosomes that are more dense. So if you can imagine, since these chromosomes all have nitrogen in the N15 form, they're going to be further down. If you put them in a centrifuge, it spins round and round and round and round. And just like a washing machine, as it spins, the heavier ones go further down. As you spin the spin dry in a washing machine, all the clothes stick to the outside. Well, these test tubes get twirled around in a machine that just spins them at incredibly high speeds, called a centrifuge. And since this has nitrogen 15, and therefore this chromosome is going to be more dense, it's going to be further down in the column than this particular nitrogen 14 chromosome. Same chromosome, just this one was built with nitrogen 14, this one was built with nitrogen 15. So let's take a look at the questions they're asking here. Why is a nitrogen label a good tool for studying DNA? The best answer is here. You see, these are the A, C, T, G of DNA, and here's the U from RNA. What is it that you notice about these that helps you answer this question here? I'm not even going to tell you, because I think you could figure that one out on your own. What other molecules in a cell have nitrogen in them? Well, that's a good question. You should be able to answer that question rather, if you just look it up. Even though you could answer that question, number 2, what other molecules have nitrogen in them, you should know that obviously proteins have them. These are amino acids that make up the protein, any protein, and different combination for each different protein. But what do you notice is in all of these? Well, nitrogen, to answer number 2. What's the difference between nitrogen 14 and nitrogen 15 at the atomic level? I already answered that for you. 15 has an extra neutron. And if you have one extra neutron for each of these, obviously, and there's a chromosome has millions of these, if not billions, of these nucleotides, then you can figure out quickly, well, probably millions. You're looking at, then you can figure out quickly that the one that has a heavy nitrogen on that makes it up is going to sink lower than one that has lighter nitrogen. So this one's going to be, the ones that have the N15 are going to be denser. What's the term for 2 atoms of the same element with different molecular masses? Well, we already talked about that. They're isotopes. And give an example of another element that has atoms of more than 1 molecular mass. Geez, just about every atom. But an example would be carbon 12 and carbon 14. And carbon actually has a bunch of isotopes, and many of the atoms have many different isotopes. It's just a matter of which one lasts longer, which one is more common, which one is less common. Hydrogen, for instance, has 1 proton and no neutrons normally, but sometimes it has a neutron. So you have hydrogen 1 or 0, and then hydrogen 1, atomic mass of 1, and hydrogen atomic mass of 2. The average is 1.00 something. I can't remember, 0.14 or something like that. So you can see that when you're looking at it, most of the hydrogen in the universe is hydrogen without a neutron, but it does exist with a neutron. Just as most nitrogen is N14, but you find N15 as well. Okay, so in part 3, they show you the results of the experiment, right? So these were grown in N15, and these were grown in N14. So these cells, these bacterial cells, were grown in a different culture with only N15. So they were all, after a couple generations, all you had were the chromosomes that have, my dog is in the background, so forgive me. She is not breathing well while she's sleeping. So N15 is much heavier, much denser, because it grew up, these cells made their DNA using N15. These made their DNA using N14. So they're less dense. After 1 generation, you ended up with all the DNA, all the chromosomes being here in the middle, in between M14 and M15. Suggesting very strongly that the material that is inheritable, that's passed down from 1 generation, is definitely DNA. DNA is definitely passed down, because some of the DNA here was N15, exactly half the DNA was N15 and the other N14. Half should be important to you, because you know eukaryotic cells do meiosis, and they split their genetic material in half. Now, these are bacteria, so they're not necessarily growing. Their material. They're not exactly exchanging their DNA half and half, but they are growing in the new material that only had N14, and still half their DNA, half the chromosome is N15, the other half the chromosome was N14, suggesting they have 1 new strand and 1 old strand in the chromosome here. Because remember, all the cells grew up with N15 as being present, then they were introduced to a medium that only had N14, but the chromosomes didn't just all turn out to be N14, did they? Half the molecule, half the chromosome was N15, The other half was N14, giving you a chromosome that had in a density that was in between the two, suggesting that you didn't have, you had the semi-conservative model. The semi-conservative model. So you had one, you kept one old strand and you built a new strand using this new DNA. So which one of the three models of DNA replication are ruled out by this experiment? Well, you're ruling out the conservative model for sure. That's gone. And probably the dispersion model. But the conservative model is ruled out because if it was the conservative model, you had one new one new chromosome that was only N14, that would be here. And then you'd have one band that was only N15 chromosomes, and they'd be here. You wouldn't have this middle bit. You wouldn't have a chromosome that was in the middle of the density. This 100% of the new chromosomes were in between N14 and N15. So that half number keeps coming up over and over again. So number 2 asks the question, what would it look like if the conservative model were the model? And I just told you a moment ago, and I'll go ahead and draw it out. What I would expect is you'd have your tube, and let me erase that. It's a horrible drawing. Someday I'll learn how to write on one of these. You'll have your tube, and it's not much better, but there it is. It's actually worse. I can't believe it's worse. All right, let's try that again. I don't have my stylus, so I'm using my finger. Forgive me. There, that's much better. Still not great. But here you have a test tube that what I would expect if you had the conservative model in play, then you would expect one dotted line at N14, one band at N14 after centrifuge, after centrifugation, right? And then you'd have another band at N15. That would make sense to me if you had a conservative model. In other words, if your chromosome after DNA replication were grown in N14, if you took these N15 cells and grew them up in N14, you'd expect that if the old ones stayed together, you'd have a band of old chromosomes here, and you'd have a band of new chromosomes with the lighter nitrogen up here. But that's not the case. What you got was this, saying that this model, this conservative model, was wrong. Okay. What could they do to tell which of the two remaining models was actually happening using the tools that's already been described? So what would you do to try to prove that it wasn't this kind of pieced together molecule that was the dispersion model, where you had like little pieces of DNA being put together? How could you solve that? What kind of experiments would you design using any one of the RNA, DNA, and proteases, using these isotopes to grow new chromosomes? How would you design an experiment that would get rid of, to prove that you don't have the dispersion model? So here we grew the cells again in N14, so only 14. Now remember, we suppose, a chromosome that has one strand made of N14 and one strand made of N15, so it's in between the N14 and N15 areas when we run it through a centrifuge. But when we look at a second generation, keep growing them, and again in N14, and then run them back in a test tube again, and what you find is that you get two bands, one at N14 and one in the middle. So why do you get this one up here at the top, the ones that only have N14? Well, remember, we're talking about semi-conservative. So when this band that has one N14 band and one N15, when they make new copies, you're going to have one that's still half N14 and N15. But the other one is going to be N14 and N14. So you'll have a band up here. You'll never have two N15 bands, for instance. And so when we think about the dispersion model, and actually the first experiment would actually also kind of move toward in that direction, is you wouldn't have two distinct bands, right? If you were just randomly putting bits and pieces together, what you would expect to see in a test tube, and this is answering the two questions below, you'd have a test tube. The S would have a band here, maybe there'd be a band here. There'd be a bunch of bands. There'd be more than two bands because it depends on randomly did you get M14s or did you get M15s? Did you get 2 or 1? You wouldn't expect this kind of you wouldn't expect this 50-50 half of a division the way you're getting here. You'd expect a bunch of different lines, suggesting again that the semi-conservative model is how DNA is replicated, where you're getting one old strand sticking with the new strand. Excellent experiment, I think, and went down in the history of biology as helping prove that DNA is replicated using the semi-conservative model, meaning one is being used as a template to build the other. So here again, you're going to go ahead and write your paragraph, a well-constructed essay paragraph that looks at how you use the diagrams to really explain. And if you use diagrams, make sure you explain each of the steps of the diagrams. But you want to describe in detail the chemical structure of DNA and how DNA is replicated. All right, so nobody's going to ask you how you do it. It's exactly how you do it. We're going to keep describing it. Even Nielsen and Stahl really didn't understand how it was done. They didn't understand the double helix. They didn't understand a lot of what was happening in DNA, but they were able to control the environment, use control groups, use experimental groups, and using hypothesis and repeating the experiment, they were able to determine that we had a semi-conservative model of DNA replication in play. So your job here is to describe what the structure of DNA is and what kind of model, when you're duplicating DNA, what kind of model actually happens, and either diagram it or describe it. And then that'll complete your case study here for chapter 7. You have a copy of it. If you don't, you have a copy of it here, but I could always email you a copy again if you need it. Let me know. Good luck.